Hello everyone, so I'm Shreya and I'll be presenting on the topic of molecular docking and lead discovery. And the topic which is provided to me was glioblastoma and the protein name is EGFR. So these are my table of contents. So coming to introduction, glioblastoma it's a highly aggressive cancer of a brain that grows and spreads very fast. It's a more severe type of brain tumor of grade 4 and EGFR it is epidermal growth factor receptor. It's a kind of receptor that is a tyrosine kinase is involved in cell proliferation as well as survival. Some of the functions that are involved in EGFR are ligand binding, receptor activation, autophosphorylation and activation of downstream signaling pathways. So coming to data collection, so databases used for searching a protein is UniProt or RCSP. So here we search for a protein of interest and then we filter it to the reviewed and the human and then we have to download it in the PDB format and we use PubChem for downloading or ligand of interest, and the top 5 ligands which I've selected is gefitinib, erolitinib, afatinib and osimertinib and lapatinib, and it is downloaded in a 3D structure in SDF format. Molecular preparation and molecular docking. So here we install PyRx software and we go to the files on the top left corner and we have to click on the load molecule, and after loading a molecule click on the right click on the protein and then AutoDock followed by make macromolecule, and we have to go to the second column of the top left side and then open variable, and then we have to insert a new item where we add the ligands, and then each ligand we have to minimize selected and convert the selected ligand to AutoDock ligand, which is we have to store it in a PDB-QT format, and then click on the Vina lizard and select local and click on the start. Followed by we have to expand. A grid appears around the sides of the protein in which can arrange or we can like adjust the grid size so that protein comes stays inside the grid. Next, we have to run docking using AutoDock Vina integrated in the PyRx, and after 100% of it is completed, we observe the below given table in which binding affinity is mentioned. It is used to find the binding affinity and also RMSD UB and RMSD LB as well. Next step is splitting into individual poses. We install Vina split and then we open the command prompt and change the directory to Vina underscore split input file name and underscore output plus out plus pdbqt. And this is a result which I obtained after I used command prompt. And now all the ligands are split properly, resulting in the each conformation of the ligand, which states that using a Vina split software, all the ligands are split individually. Next is generating visuals. So here to generate the visuals, we have to install PyMol and Discovery Studio Visualizer, which is nothing but BioVia. So PyMol it provides 3D structures and 2D interaction map, and 3D interaction map is provided by the BioVia. So this below structure, it is a 3D structure of the docked complex. It is obtained using BioVia. So this is a 2D interaction map and the 3D interaction map obtained using BioVia, and this is a 3D structure of a docked complex which is obtained using PyMol. Here the green one states the protein and the blue one is a ligand which is attached to it. It is docked to it. Coming to the results and discussion, the best docking score obtained here was -8.3 kcal per mole for gefitinib, indicating strong and stable binding with the EGFR. and RMSD offers zero Armstrong confirms that the most reliable binding pose. The 3D visualization shows the gefitinib occupies the ATP binding pocket of EGFR, and the 2D interaction map confirms hydrogen bond and the hydrophilic interaction with the key residues. Since gefitinib is a known EGFR inhibitor, the strong docking result validates our protocol and supports its therapeutic relevance in glioblastoma. Here, docking results are predicted and required experimental validation, which is one of the limitations of docking. And yeah, thank you.